Exploring Life. Chapter 8, Eukaryotes. Part 1. Last lecture, we discussed prokaryotes, the earliest and simplest forms of life. Now, we begin our exploration of eukaryotes. Let's look at the key features of eukaryotic cells. The most obvious difference is size. Eukaryotic cells are much larger, typically ranging from 10 to 100 micrometers. In contrast, prokaryotic cells are just 0.1 to 5 micrometers. That's a huge difference, tens to hundreds of times larger, depending on the comparison. But more important than size is a key innovation, a complex internal architecture. Eukaryotic cells have a nucleus, a vast endomembrane system, and specialized organelles including mitochondria and, in some cases, chloroplasts. These features give eukaryotic cells remarkable functional diversity and efficiency. Eukaryotes emerged approximately 2.2 billion years ago, evolving from simpler prokaryotic cells. This marked a major turning point in the timeline of life on Earth. According to the endosymbiotic theory, eukaryotes evolved from ancestral prokaryotes through two major steps. First, membrane proliferation. This led to increased cell size, multiple membrane folds, the ability to engulf food particles, and other cool new features that gave the cell more complexity and flexibility. Second, the acquisition of mitochondria and chloroplasts through endosymbiosis. The membrane proliferation led to formation of the endomembrane system, a major evolutionary advancement in eukaryotic cells. These internal membranes allow the cell to compartmentalize its activities. But why is that important? Imagine a school with no internal walls. Could multiple classes be taught at the same time in that kind of space? Probably not. Without separation, lessons would overlap and everything would become chaotic. In the same way, internal membranes in a cell create distinct microenvironments, allowing specific processes to happen without interference. Can you think of your own analogy to help visualize how the endomembrane system works? What kind of system, real or imagined, helps you picture how different processes can stay organized, efficient, and well-coordinated? Several organelles are formed by the endomembrane system, including the nucleus, the rough and smooth endoplasmic reticulum or ER, the Golgi apparatus. Together, they form an interconnected network within the cell. Some components are directly connected by shared membranes, while others communicate through vesicles that transport materials between them. The membranes of the rough ER, smooth ER, and the nuclear envelope form a continuous, connected structure. Other organelles in the endomembrane system are linked indirectly through vesicles, tiny, membrane-bound sacs that transport materials between compartments. For example, vesicles bud off from the rough ER carrying newly made proteins to the Golgi apparatus for further processing and packaging. From there, Golgi-derived vesicles deliver the final products to their destinations inside or outside the cell. After the endomembrane system was established, the next major step in eukaryotic evolution was the acquisition of mitochondria and chloroplasts. According to the endosymbiotic theory, these organelles were once free-living bacteria. As we discussed in the last lecture, some prokaryotes already had impressive abilities. Aerobic bacteria could carry out respiration, and cyanobacteria could perform photosynthesis. The ones that were about to be engulfed were probably very similar. The future eukaryotic cell likely intended to digest them, not exactly the friendliest beginning. But instead of being broken down, these bacteria stuck around, proved incredibly useful, and eventually became mitochondria and chloroplasts. They've been part of the cellular team ever since. This image illustrates the two major events of endosymbiosis. The first endosymbiotic event was the acquisition of aerobic bacteria capable of respiration. These bacteria were engulfed and eventually became mitochondria, powering cells with efficient energy production. The second endosymbiotic event involved the engulfment of photosynthetic cyanobacteria, which later evolved into chloroplasts. Mitochondria were acquired by all eukaryotes, so every eukaryotic cell has, or once had, mitochondria. Only photosynthetic eukaryotes, such as plants and algae, have chloroplasts in addition to mitochondria. The illustration shows a generalized eukaryotic cell and its main structural components. The cell membrane, the cytoplasm, the nucleus, and various organelles, including mitochondria. It might look a bit complex at first, but don't worry. We'll make sense of it. The major vital processes in eukaryotic cells include the manufacturing and breakdown of molecules, the processing of energy, and providing structural support and movement. Each of these functions is carried out by specialized organelles, working together to keep the cell alive and running efficiently. In the next portion of the lecture, we'll take a closer look at each of these essential processes. Let's start with the manufacturing and breakdown of molecules. Eukaryotic cells have advanced systems for protein synthesis and processing, including mechanisms to ensure proper folding into tertiary and quaternary structures, a task supported by the endomembrane system. At the same time, these cells manage precise breakdown and recycling, allowing for the efficient digestion of molecules and the reuse of valuable components. Key organelles involved in the manufacturing and breakdown of molecules include the nucleus, the nucleolus, ribosomes, the endoplasmic reticulum, the Golgi apparatus, and lysosomes. The nucleus is the central organelle that houses the cell's genetic material, DNA. It contains DNA sequences that serve as blueprints for protein synthesis. The nucleus also regulates cell activities and ensures the maintenance and inheritance of genetic information. The nucleus is enclosed by the nuclear envelope, a double membrane that protects its contents and separates them from the rest of the cell. Inside, it houses chromatin, a complex of DNA and proteins that helps organize and condense the genetic material. This electron micrograph shows the nucleus, the nucleolus, the rough endoplasmic reticulum, and other internal structures within a pancreatic cell. These components work together in the synthesis and processing of proteins, a key function of cells with high secretory activity like those in the pancreas. The nucleolus is located within the nucleus and plays a crucial role in assembling ribosomes, which are essential for protein synthesis. Its main function is to produce ribosomal RNA, rRNA, and combine it with proteins to form ribosomal subunits. These subunits are then exported to the cytoplasm, where they become functional ribosomes. Ribosomes act as molecular machines that build proteins based on genetic instructions. Because of their RNA-based structure, ribosomes are considered molecular fossils, tracing back to the RNA world, when early protobionts likely used RNA not just to store information, but also to catalyze chemical reactions. In modern cells, ribosomes are assembled in the nucleolus and then exported to the cytoplasm, where they carry out protein synthesis. Cells with high protein demands, like those in the pancreas or muscles, contain large numbers of ribosomes, reflecting their vital role in keeping the cell running. There are two types of ribosomes in eukaryotic cells, free and bound. Free ribosomes are dispersed throughout the cytoplasm and synthesize proteins used within the cell, such as cytoplasmic enzymes. Bound ribosomes are attached to the endoplasmic reticulum, forming what we call the rough ER. These ribosomes synthesize proteins destined for secretion or for insertion into cell membranes. The endoplasmic reticulum, or ER, acts as a major biosynthetic factory within the cell. There are two types of ER. The smooth ER is responsible for lipid metabolism and detoxification of harmful substances. The rough ER, studded with ribosomes, is involved in the synthesis of proteins that are destined for secretion or integration into the cell membrane. The Golgi apparatus is made up of a stack of flattened, membrane-bound sacs. It even looks a bit like a stack of pancakes. It receives, modifies, and sorts materials that arrive from the endoplasmic reticulum. These materials are carried in by transport vesicles, which deliver them to the Golgi. It acts as a central hub for modifying, packaging, and directing cellular materials to their final destinations either inside the cell or for export. Think of the Golgi apparatus as a sorting and shipping center in a factory. In this image of a white blood cell, the Golgi is visible as a stack of semicircular, flattened rings. You can also see several vesicles nearby. These are responsible for transporting materials to and from the Golgi apparatus. Now, take a moment to think of your own analogy to help visualize how the Golgi apparatus works. If the Golgi is responsible for sorting, modifying, and sending off materials, what does that remind you of? This image summarizes how key organelles work together in the manufacturing and processing of molecules. The nucleus provides the genetic instructions. Ribosomes use those instructions to build proteins. And the endoplasmic reticulum helps with folding and initial modifications. From there, proteins are sent to the Golgi apparatus, which acts like a sorting and shipping center. The Golgi directs these substances in three main ways. 1. For export outside the cell. 2. For internal use, or 3. To form lysosomes, which handle waste breakdown. This coordinated pathway ensures the cell runs smoothly and efficiently. This image helps summarize how key organelles work together to manufacture and process molecules. The nucleus provides the genetic instructions. Ribosomes use those instructions to build proteins. And the endoplasmic reticulum, ER, assists with folding and initial modifications. Next, proteins are sent to the Golgi apparatus, which functions like a sorting and shipping center. The Golgi directs materials in three main ways. Export outside the cell. Delivery for internal use. Formation of lysosomes for waste breakdown. This coordinated system ensures the cell operates smoothly and efficiently. Lysosomes play a key role in maintaining cellular health by breaking down and recycling excess materials, pathogens, and damaged organelles. Here's how the process works. First, unwanted materials are enclosed in a membrane-bound vesicle. This is endosome formation. Next, the endosome fuses with a lysosome, forming an endolysosome. Then, lysosomal enzymes digest the contents, and useful molecules are recycled for reuse. Finally, any undigested remnants are expelled from the cell, keeping the internal environment clean and balanced. In this image, we see cellular digestion in action. Particles are first enclosed within an endosome, which then fuses with a lysosome. Inside, lysosomal enzymes break down the contents. Any leftover, undigested material is gathered and eventually expelled, helping keep the cell clean and functional. But here's the twist: not all cells are great at taking out the trash. Neurons, the cells of your nervous system, are especially bad at it. Over time, they tend to accumulate waste, bits of cellular debris the lysosomes couldn't break down. As you age, this buildup increases. Scientists are actively exploring whether this process can be reversed, and if helping neurons clear the clutter could improve brain function. But for now, one thing's clear. Your neurons get more constipated with age. And no, prune juice won't help. Exploring Life Chapter 8. Eukaryotes. Once again, it's time to pause and review. Let's check your understanding with a quick self-evaluation. Take a moment to reflect on what you've learned so far. Can you explain the key structures and functions of eukaryotic cells? Do the analogies and examples make sense? Use this opportunity to reinforce your knowledge before we move on.